their methods use machine learning to optical or to acoustic imaging, and their main goal is translation of different techniques and technology to clinics. So thank you very much for being here, and we look forward to listening to your presentation.
And thank you very much for that very nice introduction. Yeah, so there are no more words I need to say about us, but I'm going to talk, and we are also going to talk about computational biophotonics and our work translating optical technologies to surgery. We're doing this with computational methods, obviously, because we're a computational division. All right, so as I said, the objective of the whole thing is we have a lot of nice biophotonics techniques that give us very nice images preclinically. Now we want to move this into the clinic and see if we can actually do some interventional stuff with it. And for that, we will use a lot of multispectral techniques, meaning we will use specific bands in a wide range of wavelength where we can distinguish between different chromophores. Mostly it will be about blood, so just as a precursor to the whole thing, hemoglobin and oxyhemoglobin are the two main chromophores that we will be distinguishing in the next slides. There's also a lot of other stuff you can do, like FET and whatnot, but for this, it's those two. And with this, so the two technologies that we will be discussing in depth is multispectral imaging, so surface imaging with multispectral cameras, and then deep imaging with multispectral photoacoustics is my topic, and I will be talking about that later. But to start off the whole thing, Leonardo will give us a bit of context and a bit of different techniques that also exist by photonics.
Hello everyone, thanks for that introduction, Thomas. So as he mentioned, I will be talking today about multispectral imaging, but surface imaging. But first of all, let me talk about different techniques that we are currently evaluating. So first of all, what all of these techniques have in common is that they exploit one specific kind of information, and this kind of information is how light travels inside the tissue. So once the light travels inside the tissue, there are three main interactions that it can go through. One of these is it can suffer specular reflections, it can suffer from absorption or diffuse reflections. And most of these techniques only look at diffuse reflections. And this is because if you look at the spectrum that you get with different techniques, you can see that, for example, depending on the content of blood or the content of oxygen in the tissue, the spectrum will look different. And now the main goal of each of these techniques is basically try to associate each of these spectra with what the content of these physiological parameters are. So first of all, let's talk about a couple of these techniques. So the first one is optical coherence tomography, and you might have heard of this because it's basically widely used in ophthalmology. And one of the nice characteristics of optical coherence tomography is that basically what you can do is it can give you a very, very nice contrast of the different kind of tissues that you have, and it can also give you an idea of what the content of oxygen and the flow rate of blood is in the tissue. But not all of these techniques can give you this information, just the time domain techniques, because these have been widely studied. And this is there are, for example, Fourier domains, applications for OCT, Doppler domain also, or Doorbane, or even inverse. Now, one of the key characteristics of this technique is that it can give you a very nice resolution, about 1 micrometer resolution, which is what most of these techniques are lacking. The next is called diffuse correlation spectroscopy, and this is another technique that exploits the information in the time domain. And one of the nice characteristics of this is that if you, for example, launch a laser pulse train of waves into the tissue, what you can do is, okay, if you measure at a certain distance, then you can separate the signal that comes from the surface of the tissue and the signal that comes from deep into the tissue. And with this, you can create like a nice 3D map of what the composition of the tissue is. The next one is called polarization spectroscopy, and this basically what it does is, okay, I launch polarized light into the tissue, into, for example, skin, and then I try to filter the different polarizations that the light reflects. And with this, what you can do is basically get rid of melanin in the skin, and then you get mostly signal just from the chromophores inside of tissue, for example, hemoglobin and desoxyhemoglobin. Now this in the second image here, what you can see basically is, okay, if you have finger and then you occlude it, then what you can see here is the concentration of hemoglobin, and once the finger is occluded, you can see that you can actually measure how much the amount of blood changes in the finger. This is just an example. If you want to read more about it, you can go for the publications. And this was not the last but not least that I want to talk about is fluorescent lifetime imaging. And with this, basically what it exploits is, again, the time information of fluorophores. And with this, what you can see is basically, okay, different fluorophores will have a decreasing signal over time, but how this signal is stamped over time will change depending on the fluorophore that you have. And with this, you can basically recover the concentrations of different fluorophores in the tissue. And this is just an example of, for example, if you have OGB1 fluorophore, and then you inject it into a culture of neurons, then you can actually get a concentration of calcium inside the neurons. And this is important because with this you can actually measure how active each of these neurons are. And now, before I go forward, all of these techniques are very nice. Some of them have some advantages, some of them have disadvantages, and now we go into multispectral imaging. And why multispectral imaging? And this is because multispectral imaging can give you a wide field of view, can give you high resolution, and can be basically measurements that can be in real time. Now, what's the motivation for our work? So, for example, if I show you 4 of these kidneys, and then I ask you which of these kidneys is perfused, meaning which of these has blood supply and which one doesn't have. This is basically what the surgeon looks at, and probably you will agree that this is quite difficult just from RGB images, okay, to actually estimate which of these is perfused. And as it turns out, some of them are perfused and some are not, and even this one here is partially perfused. Now, and this is because of how the human eye works. The human eye works in the way that it has 2 receptors, mainly a red, a green, and a blue receptor. And now the way that you can think about a multispectral sensor is like if you have an eye with supervision, in the sense that you can actually record more bands, and then you will get more information about the spectrum. Now, why do we want to capture more information about the spectrum? This is because we work in the visible region. Why do we work in the visible region? This is mainly because in this region here, the main chromophores in the body are hemoglobin and oxyhemoglobin. And in internal organs, basically melanin doesn't play much of a role because there is hardly any melanin inside of the body. And so the main absorbers in this region are these 2, hemoglobin and oxyhemoglobin. So once you can estimate how much of this is inside of the tissue, then you can estimate how much blood supplies there in an organ or how oxygenated it is. Now, if you look at a multispectral recording, this is how it looks like basically. And now if you take a look at one single pixel, this is what you will get. But if you just look at this, then this will not give you much information, right? If you present this to a surgeon, this wouldn't tell him anything. So what we have to actually look at is the inverse problem. How do we go from this spectrum to actually something more meaningful, which is the tissue properties, for example, perfusion or tissue oxygenation. Now the main problem is that there is no gold standard for measuring how much oxygen or how much blood is there in the tissue, right? And how do we tackle this? We tackle this with simulations. Now what we did is, okay, we gather all the information that we can get about how tissue is composed, mainly, for example, optical properties like the anisotropy of the tissue, the thickness, how much blood there can be in different kinds of tissue, how much oxygen, etc., etc. And now, once we do this, once we have this tissue model, we construct a tissue in different layers depending on the kind of tissue that we want to study, and then we launch Monte Carlo simulations. Now, Monte Carlo simulations is nothing more than simulating how the light basically travels into tissue. Now, once we have this framework, what we can do is generate thousands and thousands of simulations. And the nice thing here, because this is actually the key method, the key fact in our method, and this is that each of these spectrum will have associated all the tissue properties that we designed at the beginning. So for each of these spectrum, we can say how much oxygen, how much blood is there. Once we have this label data, what we can do is then train a machine learning model that then from the simulations will learn to estimate in real life measurements how much oxygen and how much blood is there. Now, this is basically just to give you an idea of how the setup is. On the left, what you see is a normal laparoscope attached to an RGB camera, and on the right, what you see is again a normal laparoscope, but now attached to the multispectral camera that we have. As you can see, this is very small, very compact. It's very, very easy to use for the surgeons. Now, what you will see now is one of the first in vivo patient studies that we have, and what we see here is a kidney, and now we are clamping the main vessel. And here in the color window, what you see is the content of blood in the tissue, and you see the color changing to blue, and blue means less blood, and red means more blood. And you see how the model actually estimates quite well how the content of blood changes in the tissue. And now what you can see here is the same kidney, but now we will release the clamp, and you will see here how the oxygen changes in the tissue. And you can see how it quickly changes because all the blood rushes into the kidney. I think this is quite exciting, but so now that we have this model, there are some open questions still. So just as a recap, we have a model which we train on simulated data, and then we get an estimation of SAO2. But now what most of the machine learning models lack is the fact that they cannot give us an idea, okay, how certain are you about this estimation of oxygenation, for example. Now, it would be actually nicer to not just estimate one single value of oxygenation, but actually to estimate one distribution. Now, the main problem now is that, okay, so if we have two different spectra with two very different levels of oxygenation, and now we map this into the training spectra, what it can happen is that the training spectra actually looks very similar, and then the machine learning algorithm will not learn how to dissociate these two values. Now, how do we tackle this? We tackle this with something called invertible neural networks. And how invertible neural networks tackle this is that they encode all the information of the distribution of your training data into a multivariate Gaussian. And then if you sample from this Gaussian, what you can get is actually the distribution of your spectrum, which is this actually that you see here on the left. And you can see how two peaks actually start to appear, meaning that here I think it's around 63%, and this is around 83%. Yeah. Now, there is also another question. Okay, you have your nice invertible neural network, but this will, in theory, only work if your test data is within the distribution of your training data. But now, what will happen, for example, if you give a spectra that is completely out of distribution from your training data to invertible neural network, what happens is that there is no performance guarantee. So it will not know actually what to do with it, right? Because it hasn't seen it before. Now, how do we tackle this now? We tackle this using different outlier detection methods, and so now, for now, we have a focus only on regression and uncertainty estimations, but it would be actually nicer if we can also integrate our distribution detection, because now we can estimate, for example, okay, this spectra here actually doesn't look like tissue, so I shouldn't estimate something from this because I don't know what it is, right? So how we can think about this? So we go for ensemble methods, and just to give you a quick example. So if you have, for example, two distributions of points, and you want to classify them in two different classes. So what you can do is, for example, you can just draw a line to separate them. But you could, most likely, all of you would agree that there is different lines that will separate these two distributions, for example, this one. Now, the ensemble methods, what they take advantage is, okay, if you have a lot of models that will estimate differently how to separate these two distributions, then if you see this point here, which is an outlier, each of these classifiers will classify this point sometimes in this distribution and sometimes in this distribution. So the variance of the classification will be high for this point, which is an outlier, and will be low for these other points, which are the ones that are within distribution. And this is what we take advantage of. Now, this is an example of what we did. And this is basically just trying to estimate when the illumination changes in a scene. And you can see how the method actually quite well estimates when the illumination changes around here. Now, let's get back to the main problem. So we had a layer model, and we tried it in kidney. And that's fine, it works perfectly. But now, the question is, okay, does it actually generalize to all the parts of the body? You just tried it in kidney, but what about the brain or something else that you would like to try? So this actually went into the brain, and we tried something that is called spreading depolarization. And spreading depolarization is a side effect from stroke. And what happens is that the neurons in the brain get massively depolarized and start to consume a lot of oxygen. And this region, which is deoxygenized in the brain, starts to move in the surface of the brain as a wave. And a lot of people actually, people that have migraines, actually start to see this quite often. And this is, they see it like a black window that moves in their eye region. And this is basically one of these slits that is moving into the region of the brain that actually processes the images that you are capturing with your eyes. Now, what do we do? What we did is basically we have a pit model. We do a craniotomy, and we place the multispectral camera on top. And then we record these images, and now we estimate how much oxygen is there in the tissue. What you will see now here is on the right. This is a part of the brain, and here remember that high content of oxygen is yellow and low content is blue. And you will see on the left is just the mean value on two different ROIs, in this one and in this one. And now what you can see here is basically this brain depolarization is moving quite nicely on the surface of the brain, and you can also appreciate it here on the left when the oxygen goes up and down in each of these ROI. And this was quite exciting when we saw this, because the model was actually not trained or not designed to work on the brain. We just generated a lot of simulations based on what we know about tissue, and this actually shows that it generalized quite nicely, and we have a nice wide field of view and very high resolution with multispectral imaging. And now all of this that I have talked about is only on the surface, but now what if we want to look into the depth of the brain? And now someone will take over to talk more about this.
So exactly. So what we're also working on is multispectral photoacoustics, and the idea there is so it's nice to see all of this on the surface, but there are also some open questions to what happens in, for example, this is called gyroencephalic brain, so it has folds, and in those folds there's also gray matter that also should be depolarized, but it sometimes kind of functions as a hurdle of depolarization. It doesn't spread through the folds, and we would like to see what happens in the folds. For example, our collaborators would like to see that. And for that, we looked at that with photoacoustics as well. But to start talking about photoacoustics, I should probably explain to you what that is. So basically, again, the same spectrum, but now we're looking into the near infrared. And why that is it has better light penetration. So if you have a light source, and your light source, for example, this one is shiny and red, and if you occlude it with some tissue, you will see that the red passes quite well, and the infrared passes even better. And it passes quite deeply, so you see some light after a centimeter or two. Maybe 2 will be hard, but you still see some light. And that's nice, but why can't you actually use that to image stuff? It's because it's diffuse, and if you want to image something, you just get a blurry mess and nothing specific. So, but it still penetrates. It penetrates that 2 centimeters, and that's what we use in photoacoustics. So again, we're gonna use the same contrast and then look at the same stuff like SO2 or total hemoglobin. But how we do it is different. So in photoacoustics, you send in a laser pulse into the tissue, like this, but pulse. And that laser pulse is diffused, but still gets absorbed locally. So if there is a vessel that absorbs that laser radiation, then it will heat. It will heat suddenly, expand, and there will be an ultrasonic shock wave. And if that laser pulse is short enough and high intensity enough, that ultrasonic shock wave is measurable with ultrasound transducers on the surface. Sounds a bit far-fetched, but it actually is. I won't bother you with the calculations, but basically that's possible, and that's the principle of photoacoustics. You send in light, it gets absorbed, you get a thermal expansion, and the shock wave is measured by an ultrasound transducer. Just as a qualitative example, so this is a DKFC logo on a sponge in a milk bath. Milk bath. And then you see, you can't see it anymore, and you can scan it with an ultrasound transducer by moving over it and 3D reconstructing the whole thing. And that's how it looks like. You get the morphological information, there's some hard thing or a spongy thing. And then if you do photoacoustics, you get the additional optical information. So the value proposition is quite obvious. You get some more information with your ultrasound technique in depth. And that's exactly how this is used in brief, and how we actually, our first system, which we built in-house, more or less in cooperation with Fraunhofer and a bunch of other guys, is just that. You have an ultrasound data acquisition system, a laser, and then a bit of control software that checks if the laser pulse fires, get an image and whatnot. And then you can scan stuff with that handheld probe. And you get, just like in an ultrasound system, something like light video resolution. You can go up to 100 Hertz by now with lasers. We have 20 Hertz with that one. And resolution like ultrasound. And then you get realistic imaging depth of just about 1.5 centimeters. Some people say 7, lying, but 1.5 is really possible. And that doesn't sound like much, but for optical imaging, it's great. You usually don't get a millimeter with optical imaging, and 1.5 centimeters is quite nice. So going back to the spread and depolarization. So the same thing. We're looking at the gynocephalic brain, and as I just said, so you see those folds in the brain. These wave patterns, they follow those gyri, but they don't really cross those folds. So our partners wanted to look into those, and those gray matter folds just go 1 centimeter deep. So 1.5 is quite enough to look at that. So we just wanted to see if there's possible photoacoustics. What we did is we set the transducer optically tracked on the top of the brain of a pig, and then we scanned that in 3D and had a nice 3D reconstruction, but it's not actually that nice. You get some blotty images of total hemoglobin, so we decided to just stay in plane 1 centimeter off the midline, and then, so this is how that looks like, basically from the top. This is where the transducer was placed, this is an ultrasonic image, this is an example for a photoacoustic image at 1 wavelength. And when we did that, we also placed some electrodes, like in the multispectral imaging experiments, to get a gold standard reference. It's not a gold standard, it's more a clinical reference, how that would usually be measured, those spreading depolarizations. And then you stimulate that spreading depolarization by basically dripping a lot of ions locally, and then locally that depolarization is caused, and you get that nice model of the spreading depolarization wave. It looks like that. And when we did that, we saw exactly the same thing. Like, this is now 1 millimeter under the surface. We saw spreading depolarization moving through, and to make that a bit more visual. The video isn't really smooth, but yeah. So we stimulate and we get spreading depolarizations moving through the brain. That's actually quite nice, because those spreading depolarizations at that resolution weren't possible to image before. You can image the same thing with MRI, but with like bone sequences, because the time scale is so large, you have the time that you wouldn't usually have just to image those kinds of changes. But this is 500 times fast forward, so you have time to image it with MRI, but not in that resolution. Yeah, so basically that's what we did there. It's our initial results. You kind of see something. We're now getting a new system, and we will hopefully see a bit more nice images, but that's the first step. So going from there, we also looked at, so there was not a lot of computation involved by now. We basically did some really simple stuff. We did linear spectral mixing. We just took the different wavelengths, acquired them, and then did nothing more with them. So we wanted to look at what we can do to improve that. And what we did here is we did a hypoxia experiment where we reduced the oxygen inflow into the brain, and you really see how it's reducing, and then going back to baseline, and because we increased the oxygen again. And we also took arterial blood gas measurements to get a reference for how much SO2 actually is in the arteries, and then we segmented two arteries. These are two arterial vessels, and followed them, and you see the superficial one that kind of follows the general trend that the reference measurements that we took. We kind of get not quite quantitative but physiological estimate of SO2. But for deeper regions, that's just not the case, and that's actually expected. But it's nice to see it confirmed that there's something called spectral coloring happens in higher depth. Basically, the light that is necessary to get a low oxygenation signal down here is already absorbed in higher regions, and that whole thing is a big inverse problem that it's ill posed. You can't really solve it, but we're trying anyway. And what you still get though, even if you don't do any fancy machine learning things, is you get the change detection. Even in lower regions, you kind of see that something happens here, something happens here. It's actually also NSD caused by hypoxia. And the change detection is still possible, so it's useful in some cases, but it's not quantitative. And why that is in brief. So if you measure a sound wave from a perfect absorber, so you illuminate that perfect absorber, and you will get some light fluence, so how much light there is, basically, you will get something looking like this. And then you will get a pressure distribution, initial pressure distribution, that looks a bit like this, because more light at the surface, obviously. And then you go through all the detector geometry and the detector sensitivity, and there's also frequencies, stuff happening, doesn't matter. Anywho, the thing that you actually measure in the end with your transducer looks like this. It's a time series with some measurement points here. And now the challenge is to go from this measurement back to this, which, as is obvious, obvious, it's not possible to actually do that completely if you look at it as in a closed form, basically. Because you just don't get any information here and here. How would you know that that is not an elongated circle or some oval shape or whatever? But that's, generally speaking, the thing that we always try to get from that measured time series over all that stuff that happens with the physics in between back to the spatial distribution of chromophores. And how that is done in general is you usually do some beamforming, like an ultrasound, to get from that image to some brightness image that looks like we can look at it and interpret it. You could say that it's an estimated P0, but it's just something. And then there's also motion in between. You have a free-end acquisition, you have to correct for that, and then you do some spectral mixing, and then you get some function of those spatial distribution of chromophores, which is SO2, which is what we, in this case, are interested in. And there's a lot of methods how you can do that, and we did a lot of methods there. I won't go into detail with all of them, just a few mentions, basically. So the spectral mixing, what you saw from the last slides, is also all linear spectral mixing, just linear algebra, basically. And what we also try to do there is a method which we call learned spectral recoloring, or short LSD. And we use that to quantify that basically better. I won't go into detail with the method. It's very similar to what Leo just showed you. We simulate a lot in Silico. We have a lot of simulations, and then do a pixel-wise estimation, basically learning how to decolor deeper regions that are spectrally colored. It's still work in progress, and the paper should be out in half a year or so. But there's always already a preprint of something that we did with that, if you're interested in that. We did also some other techniques. Yeah, so but more I would say obvious and easy to see are some approaches we really needed for another sample application on the carotid artery, where we wanted to look at head and neck tumors, and basically cancers invading the carotid artery tumors, and we also wanted to use blood oxygenation there as a tumor hallmark. So we worked with that guy, Morgan Bishop, and we, as an initial step, just imaged a few healthy volunteers and did a multispectral acquisition and looked at the same thing again. So how that looks in practice in our software, in live and interventionally, is basically this. This is now just a raw photoacoustic image and it's a raw ultrasonic image. You see an ultrasound image of a pulsing carotid artery. And you see the same thing here. And what you see here is cycling through the different wavelengths. So melanin is very absorbent at lower wavelengths. So you see a very high signal for melanin at low wavelength, at high wavelength, there's a low signal. So you see them cycling through basically. And you also see that basically the interesting structure is this is the surface of the carotid artery, and we would like to unmix this, but this is moving so rapidly we can't really. So what we needed there is an easy live solution to actually stabilize that and unmix it. And just basically what we did is we looked at optical flow, which is the computer vision method, just to see motion of brightness patterns in the image. But we can't do that on photoacoustic images because the brightness patterns in these images, they change obviously because the different wavelengths, we want them to change, we want to see the different information. So what we did is we estimated the brightness patterns in our ultrasonic images and then have that flow field. So if you see that here, this is basically the estimated motion flow field applied to the ultrasound image, and it kind of stabilizes that a bit. And we did the same thing. We applied the same field on the photoacoustic images and stabilized them by that. And that was one of the methods that is really just easy to use. It's real time. It's nice. You can just apply it. And it's quite useful. Another thing that we did is also working on different beamforming methods to improve that, because as you saw, there's a lot of noise, there's a lot of background noise and clutter, and also the estimations aren't quite physiological because of that high noise floor. And what we did there is, I won't bother you with how exactly, but implement a slight variation of a nice new technique. It's called delay multiplying sound. The most commonly used beamforming technique in ultrasound and also in photoacoustics is just delay and sum. You delay the lines of your signal and then you sum over it, that's all. And we modified that a bit and got twice the contrast to noise and a bit better of actual physiological estimates. So that was quite interesting. But if you are interested in that, and if you're doing ultrasound or anything, I encourage you to look at our MITK, also our toolkit processing plugin. You can find it at mitk.org, and there's also wiki/photoacoustics. We have a few installers there. I will update them. They're 2 years old, I think, by now. But there's some good stuff in there. All of our algorithms are implemented in there. There's GPU implementation, so you can run that in real time, or you can run it really fast on your data that you acquire. And yeah, we implemented a bunch of arrays and hybridizations, and if you like, you can use that. All right, so that's in a very brief form what we did in photoacoustics, a short overview. And now to our take-home messages. Basically, what we showed you is multispectral imaging and multispectral photoacoustics, and that it can be applied to interventional use cases. I mean, we're not quite there yet. There's no clinically approved anything that we have right now, but we show basically proof of concepts, and we show that computational methods can play a big part of that and can actually help to get from all that data to get the information that the surgeon might actually need in the case. And our prime example of that is spreading depolarization in a gyrocephalic brain on the surface and in depth. And my computer is not quite good enough to actually run both of those videos at the same time, apparently, but you already saw them, so it's just a reminder. All right, so for the acknowledgements, of course, we want to thank our funders, the ISC starting grant from Bioscopy, from Leonardo Ayala, our boss, who supervised all of that work, and then all of our clinical collaborators, Santos, etc., and all of our co-workers who also worked on all the stuff we just showed you. And we want to thank you. And yeah, thank you. Any questions? Yes?
I actually have a couple of questions. Sure. The first one is so I'm not a physicist, so I may make it all very very easy looking, right? But to me, optics sounds like ultrasound.
What, the optics?
It sounds like ultrasound. So all the problems that we had in ultrasound before, you have layers, and you have reflections, you have deflections, you have reductions, and you need to compensate for depth because you have less signal down there. This all sounds like the same thing you have in the line field. How much of this could you theoretically copy? At the end of the day, it's a different wavelength you're talking about, right? So ultrasound versus light. There's obviously some other physical properties, but some of the image display, image analysis, and inverse problems seem to be like very similar.
Yes, so actually, I mean, they are. Let me just, maybe I can go back to this thing. So to be brief, yeah, I totally agree. They are very similar. Basically, if you look at the whole problem that exists, there's two problems: that acoustic problem and that optical problem. That optical problem is somewhat different to just ultrasound in, but this problem is identical basically to ultrasound field. So if you have, and the beamforming techniques are also the same. Basically, you can do all of the image reconstruction techniques that are applied for ultrasound, but you just divide the time by 2 because the wave doesn't have to go in and then out, it just comes out. So you just have to divide your time series by 2 and you can apply an ultrasonic image reconstruction. The only difference is that if you want to do it multispectrally, you also have to look at that your signal stays linear. So you can't use usually non-linear filters that give you a nicer image, but linearity gets lost. Because you want your single wavelengths to still be the same signal intensity, so you can basically combine them and analyze that. But otherwise, you can just use ultrasonic reconstruction techniques, and it works. And it's usually what is done.
Okay, so
yeah.
One other question. I listened to a presentation last week in San Diego about they were claiming they would be able to produce an MRI for 1,000 euros based on light. And it was a company called Open Boiler in New York. And I actually follow them for some years now, and they actually have shown incredible images going through the brain, through the skull, and not opening up the bone. Mm-hmm. So, you know anything about this, or? I
don't know anything about that specific company.
Principle.
I guess they're using basically what they're probably going to use is some version of diffuse optical tomography, or what is basically just put in light everywhere, put in sensors everywhere, and then you get some reflectance maps, and here you can correlate that stuff. It kind of works, and if you do a time resolved stuff, you can get very nice images, basically of light contrast. The thing is, there's a lot of assumptions going in there, and the resolution is usually really poor, like %.
So it may be something different because they say the resolution is by a factor of 10 to 20 better than MRI. So I'll show you the website.
Yeah, okay. So
I think it was an infrared base.
Is it maybe for a mouse only?
No.
Because for a mouse I can see that.
No, it was. I'll show you.
Okay. Yeah. So actually there's some work. So I'm actually quite excited about, as you said, not opening up the skull because it's usually a hassle. And you could maybe do the photoacoustics without actually doing that. There is currently, I mean, the king of photoacoustics, Leon Wang, with his 50 people and 50 million a year, I assume, he's currently building a brain tomograph, actually, a human brain tomograph for photoacoustics, and he's already already gave a bunch of talks about it. It's not published yet, but it looks like he can penetrate for 5 centimeters through the skull into the brain, which, if that is true, is cool. But it also only works on bald people, naturally bald people. You can't shave your head because the rest alone basically already destroys it. So there's a bunch of work there, but it's very early stages basically. Yeah.
Yeah. Thanks a lot for the great talk. It's nice to see what happens in the optical fields until today. I have a question to the depolarizations. I mean, it's nice to have this, for example, for diagnostics, if these depolarizations occur in the brain, that you actually know there's something wrong. But from my knowledge, for a couple of years, it was still not clear if these depolarizations harm the tissue or help in curing it. So my question is, how can you use this information for therapy?
A very, very on-point question. Yeah, the thing is, that's not known actually. I mean, it is not necessarily a good or a bad thing. So the weird thing about these spreading depolarizations is they occur in migraine patients, and they don't do any harm. They maybe have restorative purposes even, but if they occur, it has been shown by now that if they occur in stroke patients, and they always occur in patients with severe stroke, then the areas where those spreading depolarizations that are hit by those spreading depolarizations are damaged, and they are damaged more as if they wouldn't occur those spreading depolarizations. So there is some aspect of it that actually damages brain tissue, and the plan here is to basically the whole project of our clinical collaborators finding basically emergency drugs like ketamine or different gases you can inhale that stop spreading depolarizations, and finding out, that's why we're doing the Olympics, finding out of different combinations of those drugs help save tissue that might not necessarily die, maybe. A lot of maybes in there. Yeah. That's the project. Of course it's not, I mean, yeah.
That's a good start.
We have to start somewhere.
Okay, thank you.
For the classification, do you use the raw information from the multispectral images, or did you convert the data in other color spaces?
Can you repeat the question? The classification of what?
You showed some image where you have the two river outliers.
And maybe that was long. That was long. Can you close it?
Yeah, here it is. Yeah. Can you use the raw data from the camera sensor, or did you convert it in other color spaces too?
Okay, so this is just an example, right? But how this works in reality when we go, for example, for these measurements, what you do is, okay, we use something called WIPE, which is an ensemble method. And this is basically training a lot of these invertible neural networks that I talked about on simulated data. And another good thing about the simulated data is that you can transform it however you want. And especially for this, you have to transform it, for example, to the illumination that you used here, so that it actually knows, okay, this is how scenes illuminated with xenon light source look like. Once you do this, then the algorithm will predict, okay, yeah, this is good, this is what I know until now. But then if you give it something that it hasn't seen before, then it actually gives you something else, which then this here will tell you, okay, the illumination has changed. Yes, exactly this one.
Oh,
So
yeah,
you only need to you only need to train it into one of the onto the things that you know, and it automatically will learn to distinguish the things that it hasn't seen.
Okay, yeah.
Does that answer your question or not?
I think he answered it, yes.
I mean, if it's not clear, tell me and I try to elaborate.
Yeah, it's similarly clear what you've done there. I think what I pointed to is not related to it.
I think what you mean, so we did that definitely for some illuminant estimations that we explicitly performed it in a lightness color space and stuff like we did these transformations for specific tasks. But generally speaking,
Okay, yes. So generally speaking, okay, so I think I got the question now. So generally speaking, for the specific sample that I showed, it's not transformed, it's the raw multispectral recording. Okay. For other methods, like for example, as we said, for illuminant estimation, then you can transform it to the Gamut space, for example, which is quite known. But that has nothing to do with these changes in illumination, just has to do with estimating how the scene is illuminated. Okay.
So no question?
Okay. So we just stay with the.
For being here again and for the talk, obviously I missed your talk. So we have a memento. I hope you like it. It's the wine, and I think it's from Magdeburg. Yes.
It's the Halbstadt.
Yes. Exactly. Nice work. I will like it. Okay, thank you very much again. And now we have a coffee break, and then we'll come back here again in half an hour to
Coming here or coming to the wing?
here. Out.
No, but afterwards here or afterwards over in the.
I think we'll come back here again.
Oh, we'll see. Okay.
Yes. Thank you.